Welcome and thank you for joining to this webinar, Advantage of Fluorescence in Cell Counting. I'm Elvis Pérez, responsible of Genomic Lab Workflow at Bonsai Lab. Bonsai Lab is always committed to the most innovative technologies and solutions for the laboratory. And DeNovix has been one of our main representatives for several years, leader in microvolume spectrophotometry technology and in cell counting in an efficient, simple, and respectful way with the environment. Today, you will be able to learn more about our CellDrop cell counter without the slides and how to improve the efficiency of your counts. During the webinar, you can ask your questions in the chat, which we will try to answer at the end of the season. I'm pleased to introduce you Dan Schieffer, Scientific Director of DeNovix. Please, Dan.
Thanks, Elvis, and thank you for joining us, everyone. Brief overview of today's agenda. First, we'll talk about DeNovix and the CellDrop. Then we'll go into our first live demo, which will be a tissue culture count using Trypan Blue. Then I'll talk a little bit about the various viability assays that you can use on the CellDrop, how they work, and why using fluorescence improves the accuracy of certain counts. Then we'll do our next live demo. That will be whole blood and nuclei samples with AOPI. Then I'll talk a little bit more about counting nuclei on the CellDrop, the workflows that would be useful for, and the advantages of using the CellDrop for those workflows. Then finally, we'll do one last quick live demo talking about data analysis, exporting features, and then a Q&A session. So we're going to start with a quick poll here. We want to know, what method does your lab currently use to count cells? And are there any other applications you would use in your lab for a cell counter? So we'll give everybody a minute or two to fill out that poll. And in the meantime, I'm just going to start the brief overview of DeNovix. DeNovix was founded in 2012 in Wilmington, Delaware, in the United States. So we're in our 11th year now. We make products based on optics and detection technologies. Our first products were the DS11 series of spectrophotometers and fluorometers, and they came out in 2013 and 2015, respectively. And then in 2019, we released the CellDrop Automated Cell Counter. One of our core values is focusing on best in industry support. And as you can see to the side of this slide here, we've won several awards recognizing us for our great support, and we're quite proud of that as a company. Okay, so just going to bring up the results of the poll here. It looks like nobody uses fluorescence cell counting, so that'll be good. I can teach the attendees some of the advantages that they might be missing out on here. And then we have some interest in nuclei QC and antibody staining, which is also great since those would require fluorescence, especially the antibody staining. So moving into the CellDrop, that is our cell counter that counts cells without slides. And we were recognized last year, since we don't have any plastic consumable associated with this product, we received the Sustainable Laboratory Product of the Year award from Select Science. Digging a little bit more into the key features of the CellDrop, the main feature, of course, is no slides required. So it uses what we call DirectPipette Technology, which simply means that you load the sample directly into a chamber and then wipe it clean when you're done. We have the dual fluorescence and brightfield optics for easy and accurate viability analysis. We also have a variety of exporting and recording options on the CellDrop. The instrument can connect to the internet via Wi-Fi or Ethernet. So you can connect a Gmail account or any kind of email account to export your data directly off the CellDrop. You can connect it with a network folder in a computer in your lab, or you can just plug in a USB and export your data simply that way. It also has a 7-inch high definition touchscreen, and it comes loaded with what we call our Easy Apps software. So now we're going to move into our first live demo here, which is a tissue culture cell count with Trypan Blue cells. So I have some jerkat cells here. This is a T-cell, immortalized T-cell line. Bear with me for a second while I mix the sample. And I'm just going to mix this 1-to-1 with Trypan Blue. Now, quick overview of how to load the CellDrop. There is a groove here on the sapphire surface. So I'm going to put my pipette directly into that groove, bring it all the way against the top surface, and eject the cells in. And you can see the tissue culture cells here on the screen. They're already pretty well focused because since everything is fixed on the CellDrop, once you get it focused once, it kind of just remains in focus. But if you want to check, there is an autofocus feature here. We also have a variety of exposure options. Right now it's in normal, which would be 1-to-1 dilution of cells with Trypan Blue. So since everything's set, I'm just going to go ahead and hit the count button. Should take about under 5 seconds to give us an answer here. Okay, so now that it's done counting, I can zoom in on the results. And you can see all of the live cells are highlighted in green. And then what it's seeing as a dead cell is highlighted in red. And then to the left here, you have all of your most important information about the cell count. So live and dead cell, total cell counts, percent viability, and then the live and dead cells per mil, and also mean diameter, if that's important to your workflow. So now that we're done with this cell count, I'm just going to wipe the sample away. I do that simply by lifting the arm and doing 1 or 2 good wipes along the bottom surface, and the same thing on the top surface here. So now the instrument's ready for the next sample. Right, so switching back to our presentation here, just going to talk a little bit more about the features of DirectPipette Technology. That means that we have 2 parallel permanent wipe clean sapphire surfaces that form a sample chamber that you load your cells into. If that sounds very similar in function to a hemocytometer, that's because it is. So the capillary action of the pipette loading the cells draws them into the chamber very evenly, and surface tension holds that sample in place. The big difference between the CellDrop and a traditional hemocytometer is that the CellDrop has a variable height chamber that's controlled by a precision screw mechanism. So that gives us 2 advantages. One, it allows for a large dynamic range. So instead of just having a single fixed chamber height, which traditionally is 100 microns, you can do 100 microns, but you can also lower that chamber height to 50 microns, or you can raise it up to 400 microns, allowing you to essentially, on the fly, dilute your sample or concentrate your sample. So load less actual cells or load more actual cells, depending on your sample's needs. Like if it's not dense enough to get an accurate count, load more cells and get a statistically more accurate count. That precision screw mechanism also allows us to automatically recalibrate the CellDrop. If there's any kind of issue, all you have to do is turn it off and turn it back on, and it will auto recalibrate itself. So now we'll talk about choosing the right counting method for your particular sample. Depending on where your sample is coming from, choosing the optimal counting method could be really critical to get an accurate cell count. Tissue culture samples, as I just demonstrated, tend to be very clean and easy to count on the CellDrop. But primary cell samples, that would be a sample that's coming directly out of an organism, such as whole blood or, say, a lymph node sample. Traditional colorimetric stains are going to make it very difficult to count this type of sample. And that's just simply due to the presence of debris or cells of non-interest, such as red blood cells. So we really recommend that samples collected from live tissue should be counted using fluorescence-based methods for maximum accuracy. And this also includes nuclei isolated from tissue or frozen or FFPE nuclei isolation samples. So I'm just going to talk briefly about the different dyes that we've been discussing. First, we're going to talk about how Trypan Blue works. Trypan Blue is a viability stain that stains dead cells a dark blue color. The dye is able to enter into dead cells through the broken down membrane, and it binds to negatively charged molecules when it gets inside of the cell. This is very commonly used in microscopy and a very common technique for counting live dead cells for at least 50 years now. And I just want to highlight a different type of dye that works very similarly. It's called erythrocin B. Trypan Blue does tend to be toxic. So erythrocin B can be used on the CellDrop, and it's a non-toxic alternative. So that's Trypan Blue. Now talking about the fluorescence-based method, which would be AOPI. AOPI is an acronym for two dyes, acridine orange and pyridium iodide. These are both nucleic acid dyes. Acridine orange is permeable to the cell membrane of both live and dead cells. And when it enters the cells, it's excited at about 480 nanometers and fluoresces at a maximum of 525 nanometers. PI is impermeable to the membranes of live cells, but can pass through the membranes of dead or dying cells, very similar to how Trypan Blue works. When it gets into the cells, it also stains the DNA, and it's excited at about 480 nanometers and fluoresces at 632 nanometers. So in dead cells, as you might have noticed, you're going to have both AO and PI. But the light, the green fluorescence emission light from AO is absorbed by the PI dye in a fret quenching reaction. So as you see to the right here, the live cells will be stained bright green and the dead cells will be stained bright red by fluorescence dyes. When we're talking about AOPI for nuclei, the concept is very similar, except you're using it in this case to assess the extraction efficiency of the nuclei instead of the viability. So intact cells will have a green fluorescent signal again, and isolated nuclei, since they don't have the cell membrane to exclude the PI dye, will fluoresce red. Any debris from the nuclei extraction process will be ignored by a cell counter, such as the CellDrop. So again, why would you use fluorescence instead of the traditional Trypan Blue dyes? Certain sample types are very technically difficult to count using brightfield techniques. So, even if you're using an automated cell counter, you still have to set protocols for your Trypan Blue based samples. And that involves a human loading cells stained with Trypan Blue and looking at it and making judgment calls about what's a live cell and what's a dead cell. With AOPI, you just don't have that. It's very clear to the cell counter what is a bright green fluorescing cell and what is a bright red fluorescing cell. The AOPI viability assay is very simple to use, as I'll demonstrate in here just a second. It works exactly the same as Trypan Blue. You just mix the sample, your cell sample, 1-to-1 with the dye, and it's ready to use. The dyes are also readily available. We do have them available through DeNovix, but you can get them from a variety of other suppliers as well. Okay, so now I'm going to move into our second live demo where I will show power of using AOPI for some trickier samples here. So the first one I have is a whole blood sample. And I'm going to open up our primary cell AOPI app. And I have my whole blood. I'm going to combine that with the AOPI dye 1:1. So, excuse me, I'm just getting the AOPI dye out here now. Fixing it. And I'm going to load it onto the CellDrop exactly as I did the Trypan Blue. So I'm going to find my little loading groove here, bring the pipette in, and inject the cells in. And what you can see on the screen here is a whole lot of red blood cells floating around in the bright field mode. But if I switch to fluorescence, you're going to start seeing some bright green cells showing. And I'm just going to adjust the green fluorescence up here slightly. And I'm going to switch to the red channel. So I'm just checking the focus and the exposure here. And I'm going to go ahead and count this sample now that it's set how I like it. So zooming in on this sample, you can see the green and red stained nucleated cells, the red ones being dead nucleated cells, green being live nucleated cells, and then all of this red blood cell debris in the background not being counted. And again, to the left of the screen, you have all of your most important count information, such as percent viability and live and dead cells per mil. So now that we have that count, I'm going to go ahead and clean the CellDrop. Same as before. This next count, and I'm going to switch over to the Nuclei app, and I'm going to mix up a nuclei sample. And something I'd like to point out while I'm counting the sample, you may see a little bit of, it's probably lint from the tissue that I used to wipe the previous sample away there on the screen. Normal, we get it all the time. And when I load the nuclei sample, you'll see that it won't be counted by the algorithm. So you don't have to worry about getting the surfaces completely, totally clean every time. Because there's a good chance that that's just going to get flushed away, but if not, it won't get counted. Okay. So loaded the sample. And you can see that that piece of lint got flushed away when I loaded the next sample. And I'm just going to zoom in on our nuclei here. And then again, I'm going to check the green fluorescence exposure, which looks like there's not too many intact cells here. And the red fluorescence exposure, which would be our isolated nuclei. Zooming in, you can see them. I'm going to go ahead and count this sample as well. It does take a few seconds longer to do a fluorescent count, but not significantly, as you can see. So I'm going to zoom in here on this counted sample. You can see the isolated nuclei and some cells that are still intact here in this sample. Looks like the extraction efficiency was about 70%, which is a bit low, but we just prepared the sample this morning. And since it's a bit early here in the US, we didn't have time to do all the wash steps. But again, to the left, you see all of your most important information, except here you have nuclei count instead of dead cell count for the red channel, and extraction efficiency instead of percent viability. And maybe the most important number, the intact nuclei per mil here at the bottom. So I just clean CellDrop one last time. And moving back to our presentation here, I just want to talk a little bit about a study that we did comparing tissue culture and primary cell counts using these two methods, Trypan Blue and fluorescence-based methods. So we took CHO cells and cultured them, and we got mouse blood, and we counted the PBMCs in that mouse blood using those two methods. With the tissue culture cells, the serial dilutions we made showed very good agreement between the two methods, AOPI and Trypan Blue. Statistically, they were the same. But with the primary cell dilutions, we found that Trypan Blue overcounted by a factor of about 2 compared to the AOPI, and it also miscalculated the percent viability of the PBMCs within that blood sample. And this is all available in more detail on our website. It's TechNote 197. Some other applications that are really useful for fluorescence. Plant nuclei can not be done without fluorescence just due to the nature of how much debris is present in plant samples. And also plant cells can't be sequenced by single cell sequencing without first doing a nuclei extraction, as far as I know. Same goes for FFPE and frozen samples that you would then extract nuclei from. Some other applications for fluorescence would be checking your GFP transfection efficiency. We've worked with other customers who have done pollen viability counts on a fluorescent cell drop. Yeast viability and vitality can be done on the fluorescent cell drop, as can protoplast and algae counts. And we've also had some people look at trypanosome parasites, which is listed here. So I'm just going to do one last overview of the software here. No more samples, but I just wanted to point out a really nice feature of the CellDrop, and that's that all of your data is stored. So if I open up the data app here and I go into the nuclei AOPI, I can not only look at the samples that we just counted, but I can compare them, go back and compare them to some previous counts and see if there's maybe any differences, like if something wasn't quite right in my downstream processes, such as the sequencing, the data didn't look quite right, or a qPCR or something like that. You can go back, look at your data, see if anything is off, and you can also, after the fact, change the settings. So if you notice, oh no, I've been counting these samples with the wrong protocol or the wrong settings, it's no problem. You can just go back into the data app and change the settings after the fact and get a more accurate number for your counts. You also have the ability to look at various samples and graph next to each other. So over weeks or months, you can monitor your sample directly on the CellDrop, looking at things like efficiency percent or like average diameter, or if the cells are clustering more. So there are some actually really powerful and interesting features of the CellDrop for data analysis that aren't present on other cell counters in this class. So just to finish up here, in conclusion, the cell counting method you choose will depend both on the sample type and the instrumentation that you have available, of course. If you're working with mainly tissue culture cells, these can be counted with a high degree of accuracy, just using Trypan Blue and a brightfield cell counter. But if you're working with some more complicated samples, such as primary cells, or samples that might have a large amount of debris in them, such as nuclei extractions, AOPI is very likely to give you a more accurate answer. And in fact, in the literature provided by companies that do nuclei isolations or downstream processes such as single cell sequencing, fluorescence-based counting is recommended for those workflows. And I just want to point out, we do have an infographic available. So if you're interested in receiving that, it covers a lot of the information that we just covered here during this talk. In the post-webinar survey, just fill that out to receive a free copy of this infographic. It's just a nice looking thing that you can put up to decorate your lab and also hopefully give you some useful information. Okay, did we have any questions?
Thank you very much, Dan. We have several questions to answer. If you want to do more questions, you can put it in the questions and answer side under the screen.
Yeah, so I'm gonna answer the first question here. It's what fluorescent dyes are suitable with the CellDrop? So the CellDrop was really designed to work specifically with the AOPI dyes. Some other ones though that just happen to fall in the same excitation emission range would be GFP, fluorescein, and CFDA. Off the top of my head, those are the ones that I know. So if the excitation and emission for the CellDrop are available on our website, and if you want to go check other dyes, very easy to do so if you have the excitation and emission ranges of those dyes. Next question we have is, if we work with biohazardous materials, can we still use, I guess, a slide between the sapphire surfaces? Yes, actually, you can. One of the features that we built into the CellDrop is that there is a groove that the reusable surfaces sit in that will fit a slide. So not like a traditional quartz hemocytometer, but a disposable slide will fit in there if you're working, with something that's like biosafety level 2 or 3 or even higher, and really don't want to have any contact at all or have any contamination of the CellDrop. You can put it in a slide, put the slide on the CellDrop, set it into slide mode, and it will count that sample. We've also designed the dimensions of the CellDrop to fit in a biocontainment hood. So you can put it into the hood, plug it in there, and just leave it in the hood, and run all your samples on the CellDrop in there. Okay, next question. Fluorescence is better than Trypan, but is CellDrop compatible with other fluorescent dyes other than the AOPI? Yeah, so the dyes that I just mentioned, FITSI, we have one excitation channel, which is about 475 nanometer peak. And then we have the 2 emission channels, which are 535 nanometers and about 640 nanometers. I think it's plus or minus 25 or so on those. So if the dye fits within that range and it has a high enough fluorescence intensity, it should work on the CellDrop. And last question here, what is the maximum sample density that can be used? So we determine this empirically, actually, just using tissue culture cells. So, results may vary slightly if you're using something that's very small, like yeast. But we found that you can do 25 million cells per mil on the CellDrop if you have the chamber at the lower height, which is 50 microns. And that's just really because it's only imaging one focal plane, and that is about as dense as we found that you could have the cells on the CellDrop before they start just laying on top of each other, which would give you an inaccurate count since we're only doing one focal plane. And on the other end, actually, 700 cells per mil. So 700 cells per mil to 25 million cells per mil is the full range. Okay, if there's no other questions, I just want to say thank you so much for spending your Monday afternoon here with us. We do have a survey after the webinar, and we will email you a link to the recording. And if there are any other questions, please feel free to reach out to our partners at Bonsai, and their info is here.
Thank you very much, Dan. Thank you very much to join this webinar. We wish you a happy afternoon. And if you want more information about CellDrop and the other DeNovix products, please feel free to contact us. Have a nice day.